So this is a phylogeny of the major groups of all living things. We have some ancestor. So we have some ancestor to all life. We don't know what exactly it looked like. It was almost certainly a single-celled organism without a nucleus or any organelles. It probably looks something like some type of bacterium, but actually there might have been, in fact, the first life might have been actually RNA-based instead of DNA-based. And it was only became DNA-based because DNA allows for fewer mistakes in transcribing and translating. And so you're going to likely to have fewer mutations that actually make it into the next generation. And we have all these different kinds of bacteria, green filamentous bacteria like cyanobacteria, proteobacteria. Oh, here's cyanobacteria, right? And then if you go up here, there's another actual branching point. So those are nodes. And archaea, before the mid-1970s, no one even knew that archaea existed. It was thought that there was bacteria, and then there are eukaryotes, and I'll define what a eukaryote is in a second. But at first they were thought, archaea was thought to be kind of extremophiles. They were found in very extreme environments, but since then we've discovered them many other places. They are single-celled. They don't have a nucleus, like bacteria, but they have structures that are significantly different from bacteria in terms of the protein and the cytoskeleton and all that. What's interesting is that the eukaryotes are we a eukaryote? What do you think we are? Bacteria? I know, I feel like a bacteria sometimes. An archaea or a eukarya? Yeah, okay. And what do you think is different about a eukarya? We have a nucleus in our cells. So all eukarya have a nucleus, and all of them also have organelles like mitochondria. And the mitochondria are the furnace for providing the energy for a cell. So some cells have a lot more mitochondria than others. Now, and then there's an expensive intracellular apparatus for basically turning the transcript from our genomes into actual proteins, and we'll get into that as well. In addition, there are some eukaryotes that have another organelle. It's called a chloroplast. So plants have chloroplasts. And now it shows here it shows here that that animals and fungi and plants are closely related. Well, I should get a more recent one. Animals like ourselves and fungi, like our favorite truffles and chanterelles and all that, we're very closely related to each other. Now you may think, I'm not related to a mold or a fungus or a mushroom. I don't want you to disparage mushrooms. I've eaten a lot of mushrooms in my life, some that had a profound effect on me. Okay, so, but animals are much more closely related to fungi than we are to plants. In fact, plants and a whole lot of other eukarya there's two main branches, and we're in one branch and plants are in the other. And plants and a few other, so that's true. Okay, these plants actually, they should be down here. Plants and some algae like red algae and brown algae, like if you go to the beach and you see kelp on the beach, that's brown algae. They also have chloroplasts. They have chloroplasts, though, with slightly different pigment, so some of them actually look brown instead of green. But chloroplasts are the basis for photosynthetic creation or production of carbohydrates. So in other words, chloroplasts are the basis. The sun comes in, there's a whole transport chain of ions, and they actually fix carbon from the atmosphere. Namely, you take carbon dioxide, plants fix it in carbohydrates, into structural carbohydrates like the bark of a tree or the wood of a tree, things like that. And then also carbohydrates that can be used as glucose to be broken down and fuel the mitochondria. So plants also have mitochondria as well. Almost everything has mitochondria. Now, the one thing that we all share, by the way, everything that you see on here, for the most part, is single-celled. So the majority of living things on this planet are things that you actually need a microscope to see. Now, what happened was, apparently, we'll get into this more into greater depth, but as you can see, eukarya and archaea actually share a most recent common ancestor. So there was a major clade, the bacteria, on one branch, and then this other clade, including archaea and eukarya, on the other. In other words, archaea actually is more closely related to eukaryotes than it is to bacteria. But if you actually look at eukarya more specifically, there's the basis. There are bacterial ancestors that are in us. They actually make up our mitochondria, and in plants they make up chloroplasts. Okay, so it seems like there was an archaea that engulfed or was infected by some other kind of organism, a bacteria of some sort, and then eventually we ended up with eukaryota. There are some bacteria that form kind of multicellular colonies. You really couldn't call them necessarily multicellular organisms, but it's kind of the basis for that. I haven't mentioned anything about viruses, but I will. So on the left, you actually see a eukaryotic cell. And these are some of the inside stuff. We'll see this over here in a second. We have some different kinds of bacteria, staph, some of which cause disease. Most bacteria do not cause disease. Only some of them do. Some of them will only cause disease if a person actually has an impaired immune system. Otherwise, they won't. Here's a cyanobacteria. We found evidence of cyanobacteria in the fossil record from billions of years ago. And so we kind of surmised that one of the first photosynthetic organisms on the planet was something like a cyanobacteria. And they were what are called primary producers. In other words, they produce carbohydrates. They don't need to go and eat something else to make a living. Part of the process in terms of fixing carbohydrates is that they take in CO2 out of the atmosphere. They fix the carbon. You end up with water, and you also end up with oxygen being released. And in fact, over billions of years from the first forms of life that were photosynthetic, the atmosphere changed. Before then, there was very, very little oxygen in the atmosphere. In fact, oxygen was all, it's a highly reactive chemical, and it was, it formed like iron oxides and, things like that. So oxygen and some other metal or whatever, it was all fixed in that sort of form. It was not free in the atmosphere. It was only over billions of years that the release of oxygen from photosynthesis increased the level of oxygen in the atmosphere, to the point where today we have about 21% of oxygen in the atmosphere. Most of the rest is as nitrogen, and then smaller amounts of other stuff. So oxygen, now, a lot of the single-cell organisms, before there was a lot of oxygen, in fact, oxygen was toxic to it. And so not only did the buildup of oxygen was actually like a waste product, and killed off a lot of microbial life, and in fact changed the planet drastically. First of all, we had a lot of free oxygen in the atmosphere that could support multicellular life. And then eventually we got a concentration of oxygen that was high enough that it actually could support, the evolution of animals and plants and other multicellular organisms. Before there was enough, life wasn't like that at all. And then down here, we have viruses. But what I'd like to say is that most viruses are very small. They do not conduct their own metabolism. They're basically parasites. They use the cells that they infect to produce the products that they need to create new viral particles. So a virus is usually either an RNA or a DNA strand that codes for a few proteins for the protein coat, and that surrounds the virus. And then there are receptors on different cells that the viruses enter, and they actually take over the machinery of the cell to produce the different parts of a virus. The thing is, recently they found these mini viruses and macroviruses that are up to the size of a bacteria, and they don't have just like 8 or 10 or 20 different genes. They have thousands of genes, and so there's a whole question of whether they're actually viruses in the sense of what we consider viruses, and that a lot of them actually have genes that look like they carry out some metabolism on their own. Okay, let's.
Staph a virus?
What?
Is staph a virus?
No, it's a bacteria.
Oh, okay.
So there are some viruses, that are about the same size, but most of them are much smaller. In fact, you can't actually see, with a light microscope, you can't really see a virus. Well, there's some new ones out, but for the most part, it's electron microscopes and stuff. So Louis Pasteur, who was one of the formulators of germ theory, he was working on rabies, which he knew he demonstrated was an infectious disease, but he couldn't find there was any bacteria. Well, it was a virus. And eukaryotic cells are usually much larger than either. Among all living things, what's the biggest cells? Eukaryotic cells. Now, for the longest time, and still, people often refer to bacteria and archaea as prokaryotes. In other words, they don't have any organelles, they don't have a nucleus or anything like that. But it's actually a prokaryote. I guess you could use it as kind of a layman's term to describe a class of organisms that are really not necessarily related to each other. So let me go back. Okay. Now, it's considered, the only form of what we consider life are organisms that are made of cells, whether they're bacteria, archaea, or eukaryotes. A lot of people do not consider viruses actual living things because they do not have their own metabolism. However, they evolve just like any other life form. They're made up of the same genetic material as any other life form. So I actually think that a virus is actually a perfect example of an obligate parasite. It needs a host to actually do its business. So it's cut out the whole idea of needing a whole cell. Why carry around a whole cell and all the machinery that goes with it when they can just infect the cell and get the cell to do all the work for them? Now, so that's my opinion. You'll read a lot of textbooks where they say, no, it's got to be, but it's phooey. Okay? Now, all cells are surrounded by what's called the plasma membrane. And we have, this is inside the cell, hold on. Inside the cell, then we have outside of the cell. We have all these different kinds of membrane proteins that allow nutrients and ions and all sorts of material to be transported from outside to inside and inside to outside. Now, there's also a very important component of cells, and that's what's called a plasma membrane. And the plasma membrane is this area. Let's go. Is this area in between. And it's actually made up of a phospholipid bilayer. Everybody? Phospholipid bilayer. Good. Yeah. Potato. It's a fat. Now, what happens is, the reason that's important is that the plasma membrane keeps out things that are outside the cell and keeps things inside the cell that are inside, unless it's actually, there's these that are things that allow transport between the two, including water and things like that. Now it does this because it has a hydrophilic, hydro is water, philic, I think it's loving or something like that, part of the layer, which is the outside, which is actually permeable by water, and then an inner where you have these these kind of hydrophobic lipids. Lipids are kinds of fats. And they actually keep the outside stuff from getting in and the inside stuff from getting out because they're actually hydrophobic. So anything with water in it, it keeps things apart. So let's see, the hold on. Okay. Yeah, made a hydrophobic tails orient outside the membrane, away from the water, exactly. So it's here's the membrane on the inside. Is the hydrophobic part. Okay. Now, we're getting to the difference between prokaryotes, archaea, and bacteria, even though archaea are more closely related to eukaryotes than they are to bacteria. But we take a look at both. Okay, so bacteria and archaea do not have subcellular components, organelles. In other words, they don't have a nucleus. They don't have lysosomes. They don't have, let's see here. Where are they? The nucleus, ribosomes. They don't have ribosomes. They don't have mitochondria. And they don't have chloroplasts. So this is a eukaryotic cell. This is a bacterial cell. It only has, the DNA is actually all inside the cellular cytoplasm and not in a nucleus. It's also got these flagella. Not all bacteria have flagella, but many do. It's a way of propulsion. They still have a phospholipid bilayer. They still carry out various metabolic processes because they have metabolism. They are cells, but they don't have the kinds of structure that you see in a eukaryote. Okay. Now, so I mentioned that our eukaryote up here actually has all these different things in it, including nucleus, including microfilaments, including ribosomes, mitochondria, rough endoplasmic reticulum, Golgi apparatus. That's all for making various proteins. So instead of in bacteria transcribing from DNA to a messenger RNA and then taking that messenger and finally translating it into a protein, all takes place inside the cell. In eukaryotes, the transcribing from DNA into RNA takes place inside the nucleus. This RNA transcript, it's called messenger RNA, there's different kinds of RNA, then gets a long strand, it's taken outside of the nucleus, and it goes into the cytoplasmic reticulum and the ribosomes, which then translate it into a protein. So transcription and translation are now separated in different parts of the cell. And that has a profound effect. It leads to more efficient processes happening in terms of each. There's also trade-offs too. It may take longer for that to actually happen. Now, whether it's a single-cell organism like a bacteria a eukaryotic cell that's part of a multicellular organism with different tissue types. There's DNA for actually, it's not exactly a blueprint for the whole organism, because DNA doesn't like, oh, this is how it's written out. It's actually an interaction between the DNA and the environment that actually gives you the outward appearance, like human beings, right? Okay. But each of our cells has the full instructions for all of us, the whole complement, whether it's from the developmental genes, in terms of how we develop in utero, and then throughout life, and all the different processes that go on. Some of these processes are like housekeeping genes that all cells utilize. Others are only expressed in certain cell types. So there are certain proteins that are found only in muscle cells, and they're only expressed, those genes that are responsible for those are only expressed in muscle cells. The other ones are essentially turned off. So even though I may ask you, let's say, something like the genes for an eye, let's say our eye, in terms of all the different proteins that are necessary, they're only found in eye cells? Are they found in other kinds of cells? Or are they found in all cells? Well, the answer is all cells, because each cell has a complete genetic genome in it. Okay, it's not like an eye cell only has the genes that are used to make an eye. So each of our cells has the complete genome. There's something like 21,000 genes. And then there's a lot more going on there that we'll get to later. If you think genome size might translate into being special. Well, corn has about 120,000 genes. Okay, so if you're thinking that corn is more highly advanced, we'll say, than we are. How's that? I certainly like to eat it, but that's besides the point. Okay. So I just want to make sure you understand about the fact that each cell in our body has the whole genome, and not just the genes for that specific cell type. Okay. Now, it turns out that the genome, DNA, the DNA itself really doesn't do much, except it lays out which proteins are going to be made. We have something like 3 billion base pairs in our genome. In other words, the actual genome that is only producing proteins is a minuscule part of it. There's a lot of other stuff that's going on in our genome. Some of it is, we'll call them pseudogenes. They were genes that at one point were active in transcription and translation and making proteins, but now they're pseudogenes, they don't do that anymore. There are genes that are able to copy themselves throughout the genome. They're called jumping genes. We'll get to those as well. There are control regions that don't produce proteins, but they control gene regulation. Now, proteins, though, do all the work. So like I said, DNA doesn't do any work. All it does is code for proteins that then do all the work. There are proteins for everything. Hormones. Let's say insulin, gonadotropin hormones, thyroid hormones, sex hormones. Those are all proteins. They interact with other proteins, and they interact with other genes. affecting how those genes are actually expressed. Adrenaline is another hormone. Okay? Our adrenaline, let's say we're startled, it's upregulated, there's a lot of adrenaline. It turns out a whole bunch of other genes in our system. It ramps up our metabolism. A whole bunch of different things happen. Now, proteins, because we're all made of proteins, our cells have many different kinds of proteins. On the outside, they have receptor proteins that allow a cell to signal from one cell to another, usually through another protein, like a cell signaling protein, about what's going on, talking to its neighbors. There are transport proteins. There's cytoskeletal proteins. Inside a cell, if there wasn't what's called a cytoskeleton, it would just be a blob. Okay? So for cells to function, depending on what they do, there's different cytoskeleton proteins. There's antibody proteins. This is part of the immune system. So our body, our immune system, our adaptive immune system, creates these antibodies. We have white cells, which are leukocytes and things like that. T cells and B cells that are part of the adaptive immune system, that when they come into contact with something foreign, they recognize it, they glom onto it, and then they start making antibodies that are specific to it, and then the antibodies basically, anytime an antibody comes across, let's say a bacteria, there'll be tons of antibodies, and then a white cell will come along and eat it. There's these receptor proteins, as I mentioned. They receive signals and alter cell function. All cells have receptor proteins. Not all the same proteins, obviously, but specific to each cell type. And then there's another kind of protein that's very important, and these are enzymes. All they do is they're like a catalyst for a chemical reaction. Usually, chemical reactions have they have to have an energy potential to get over a certain amount of energy for the actual reaction to continue. A lot of enzymes, what they do is they lower that required energy to start the actual reaction. So, in other words, these reactions normally would be few and far between, but they're catalyzed with a protein. This is an enzyme that allows all these reactions to happen very quickly. Enzymes catalyze chemical reactions within our bodies and also outside of our cells, inside and outside of our cells. So alcohol dehydrogenase is an enzyme. Anything with an ACE, ASE, signifies an enzyme. Anytime you see ASE or NEASE, that signals that this is actually an enzyme that catalyzes a reaction. We have an enzyme that allows us to break down milk, lactase. As part of the process for making copies of our DNA when cells divide, there's a DNA polymerase. It's an enzyme. It's a protein that allows different DNA base pairs to be added on to an expanding chain. Now, a protein is made up of what are called amino acids. There are 20 amino acids. There's actually more than that, but there are 20 that life uses. And they're all isomers of a specific time, so there's something called chirality, whether it's right-handed or left-handed. So I think it's all left-handed. But anyway, amino acids make up proteins. Now, what actually codes for amino acids? The DNA is translated into amino acids, which are then strung together. And this happens in the ribosomes, outside the cell, in a eukaryotic cell, outside the nucleus, rather. So you're not going to have to memorize what goes into an amino acid. But in general, you've got this central part with an N, which is a nitrogen. Here, hold on, actually. Here we are. Okay, so you've got your nitrogen. It's there's two hydrogens attached to it. Then there's a single carbon chain, and then another carbon chain with an oxygen and a hydroxyl. And you also have another H, and then usually, and this is, so here's the amine group, here's the carboxyl group, and this is a side group. And they're strung together, but they also have a 3D structure, so that parts of proteins are accessible for reactions to happen. Whether they're actual structural proteins, like in, let's say, a cell taking in glucose, or an enzyme catalyzing that reaction. So that's the general form. You don't have to memorize that. I just want to get you an idea of what it looks like. Now, when you start stringing together amino acids, between each one, you make a peptide bond. Okay? So, now here we are again. This is this group that we looked at over up here. We've got the amino acid group, the hydroxyl group. Depending on what this is, like this is CH3, what is that? Oh, I don't know, I forget. Anyway, so a valine, which is one amino acid, looks like this. A alanine looks like this. A phenylalanine looks like this one over here. And they're all connected by these peptide bonds.
I have a question.
Sure.
Did you say that ribosomes are what make amino acids?
Yeah. So what happens is you've got this transcript that was made in the nucleus of a eukaryotic cell. So the DNA is transcribed into a single-stranded RNA. And then along this RNA, there will be corresponding, the other side chain, like a DNA is a two-stranded. So you'll always have, let's say, cytosine and guanine, which are two base pairs, and alanine and thymine that go together. Now these are DNA base pairs, okay? And so you can always see the complement in the RNA strand, because if you have, let's say, a cytosine, you know the complement is going to be a guanine, okay? So that's taken out of the nucleus, and then the ribosomes is actually fed into the ribosome, and the ribosome adds a complement to that. They know exactly what it's going to take to make a certain amino acid. Now, an amino acid is always 3 base pairs long. We'll look at this in a second. So to make a specific amino acid, one of the 20, you have to have a 3 base paired sequence. And it's either going to be there's 4 bases, There's cytosine, thymine, I think it's actually adenine and guanine, okay? We're going to look at this so you don't have to just put it, you don't have to remember this right away. We'll look at it. And a 3-base pair sequence is called a codon, and it codes for a specific amino acid. Okay. Oops. Okay. And then here's our protein. Each one of these is an amino acid, different kind of amino acid, one of the 20. You can also see that there's some structure here to this protein. You can actually have proteins with the same amino acid sequence, but they're folded differently. And one of the big questions has always been, what actually determines how things fold? Well, part of it has to do with biochemistry and whether there's a polar charge of positive or negative, things like that. But it's still actually being worked out. So a protein is folded in a 3-dimensional structure. And then, let's say it's a receptor protein on the outside of a cell. There'll be an active part or substrate that signaling proteins can then attach to. Okay, so this is the final product. Oh, a polymer is many amino acids strung together. Okay? So, some proteins are very long. Others are pretty short. And they all do things of one form or another. So, first is a protein. Now, we need protein because they're essentially the building blocks for protein synthesis, which we went on about our amino acids. So we get some amino acids, the essential ones from our diet. We have to have it from our diet. We can't make it ourselves. All different kinds of things have protein in them, including plants as well as animals. Here we have, like, you know, well, it's not, that's certainly not a ribeye. If it was a ribeye, I'd pay more attention. Ribeyes really are the best. And the worst thing you can do is overcook a ribeye, really. It's actually, a ribeye has a lot of marbling in it, which means a lot of fat mixed through it, so it gets very tasty. And then you have to do it just as sort of ambulatory in terms of cooking. You know, there's still got to be some moo left into it. But as I mentioned, there's proteins in all sorts of different things. And that's, you can actually turn proteins into glucose, but it's a very long, involved process. We'll get to it. It's not something that normally happens. So, it turns out, like I mentioned, we can't make all of our own amino acids from scratch. We do make many from scratch, but there are some that we can't. The amino acids that we can't make on our own are called what? Well, they're called amino acids that we can't make. No, they're essential amino acids. And these are the ones that we actually can't make. So we can make 13 out of 20 amino acids from scratch. But the other 7 are called essential amino acids, and we must get them from our diet. So on the left, we see non-essential: alanine, asparagine, aspartate, cysteine, glutamate, glutamine, glycine, proline, serine, tyrosine. Histidine is essential, isoleucine, leucine, lysine, threonine, tryptophan, our favorite, valine, and then conditionally means that if you don't have some of the right building blocks you might need to get those from your diet as well. So essential ones are the ones that we actually need to get from our diet. Non-essential ones are the ones that we can make on our own. Oh, yeah. Just know the difference between what's an essential amino acid and a non-essential amino acid. You don't have to memorize these different amino acids. Have I memorized those amino acids? No. Why would you do that? You can go on the internet and look it up, okay? There's better things to do with your brain than to memorize a bunch of stuff. Even though I know this is a 100 level class and I expect, you have to memorize stuff. It's too bad. The main thing is actually learning how to find the information you actually need. Okay. Nerd alert. Okay. So, who is aware of tryptophan? Who isn't aware of tryptophan? Okay. Someone who's not aware of tryptophan, where do you are you supposedly often come in contact with tryptophan?
Turkey.
Turkey. Thanksgiving turkey, right. Okay. So there's this, now tryptophan is used to make various things, a neurotransmitter, like these are basically like hormones in the brain. They're made of proteins, okay? So tryptophan is an amino acid. It's used in the production of serotonin, which is a neurotransmitter, melatonin, a hormone, another one that affects sleep. So, and then of course, during the test, of course, I'll probably ask you to completely map out what tryptophan looks like. I want you guys to have like what is that called? A nightmare that turns into a real nightmare. Okay, no, you're not going to have to do that. So the tryptophan question is, now tryptophan, remember it says melatonin is a hormone that affects sleep. You dose someone with extra melatonin and they usually get sleepy. So you know how it is on Thanksgiving. You just stuff yourselves to the gills and then you stuff some more in. And hopefully you are not a bigot against meat, like some people. And you eat this turkey. And it supplies an extra dose of melatonin. And that's why after a big turkey dinner, you all kind of tend to doze off to a wonderful somber sleep. Okay, now, what is this? When I say that tryptophan is responsible for sleepiness, and turkey has a high level of tryptophan, and that's why people get sleepy. What is that called when we say that statement? Hypothesis, exactly. Oh, I love you guys. Okay, so that's the hypothesis. We eat turkey because turkey makes us go to sleep because it has a high tryptophan level. Now, how would you test this?
Turkey.
Okay, you look at turkey and you see how much tryptophan is in it. What else? Anything else?
You can conduct an experiment and use a control group which does not eat turkey and experiment with.
Okay, you can certainly do that. What about anything else you can do in terms of levels of tryptophan?
You're just asking if you can test if tryptophan causes more sleepiness, right?
What's that?
You're just saying if tryptophan causes more sleepiness, not if turkey is.
Yeah.
That's my thought. So,
Well, let's say just right now, we're interested, does turkey have lots of tryptophan and is that responsible for sleepiness? Compared to, let's say, other
Right. So,
meats,
So
right?
you could have like the control group is eating
Right,
meats,
exactly.
of certain varieties,
Uh-huh.
and you measure their sleep.
Yeah.
And then the other group
Yeah.
is eating lots of turkey.
Right. Then the other thing you can do, of course, is just measure different kinds of foods and measure the tryptophan in each food. Okay? Now, so the prediction is that turkey has an inordinate amount of tryptophan in it. And we can take for knowledge that, in fact, tryptophan is the basis for melatonin production, and that does affect sleep. So there's a connection there. But the main thing is that turkey has an inordinate amount of tryptophan. Okay, so here's our observation. Tryptophan is a precursor for sleep-inducing hormone. I always get sleepy when I eat turkey. Tryptophan in the turkey causes my sleepiness. Prediction: if turkey causes sleepiness, then it should have higher tryptophan levels in other foods that we eat. In addition, you can also say, if I eat turkey compared to, let's say, other meat, and that's all I eat, you can, not all the other things that go into a Thanksgiving dinner, that turkey will most likely make you sleepy compared to other ones as well. But we're just going to look at what's inside. We have protein in the food. So egg white dried, spirulina dried, cod. These are all dried foods. And you have this much grams per 100 grams of food. So eggs have basically 81 grams per 100. In other words, 81%. Let's see, pork chop, only 19.7. There's a lot of fat in it. Chicken, 20.85. Okay. Tryptophan per 100 grams. And turns out in egg white you have basically 100% or 93%, 70%, right? But if you actually look at the amount of protein in each one, you see eggs is 1.23 grams of protein, or protein percent. Let's see, sesame seeds, 2.17. Sunflower seeds, 1.74. Where's my turkey? Oh, there it is, turkey, 1.11. Okay, turkey right here. Is that a lot of protein, a lot of tryptophan or not? No. I'm sorry, you guys. You don't get sleepy from tryptophan. You get sleepy from overeating. Of course, to digest properly, as you all know, you must imbibe alcoholic beverages. Okay? Now, so I just want you to remember that. Don't blame it on the tryptophan. Okay, tryptophan is a precursor to melatonin. I always get sleepy. Tryptophan in turkey causes my sleepiness. If turkey, then it should have higher levels. Turkey does not have high. Okay, so what's an alternate hypothesis, right? And we kind of just suggested two. One of them is overeating, and the other might be alcoholic beverage. And then, of course, there's that synergistic effect. You mix them together, and you get a super sleepiness that overcomes you. And the relatives have to make you sleep on the couch. Okay, let's take a little break.